Hello students, in this session we are going to understand transcription. We will understand the transcription unit, template strand and coding strand.
Transcription is the process where RNA polymerase binds to a specific region of DNA and synthesizes RNA using one DNA strand as a template. If both strands act a template, they would code for RNA molecule with different sequences, and in turn, if they code for proteins, the sequence of amino acids in the proteins would be different. Hence, one segment of the DNA would be coding for two different proteins, and this would complicate the genetic information transfer machinery. If both strands were copied, two complementary RNA molecules would form a double-stranded RNA, preventing translation. Now let us understand the transcription unit. A transcription unit is a segment of DNA that is transcribed into RNA. It consists of three main parts: the promoter, the structural gene, and the terminator. The promoter is the starting region. The structural gene is the coding region that produces RNA, and the terminator marks the end of transcription. The promoter is a DNA sequence where RNA polymerase binds to initiate transcription and determine direction. As RNA polymerase moves along the structural gene, it reads the template strand and synthesizes a complementary RNA strand. The RNA is always synthesized in the 5' to 3' direction, meaning nucleotides are added only at the 3' end of the growing RNA chain. Now observe carefully. The template strand is the DNA strand that is actually read by RNA polymerase. The coding strand is not used for synthesis, but its sequence is similar to the RNA sequence. The only difference is that wherever DNA has thymine, RNA contains uracil instead. All the reference point while defining a transcription unit is made with coding strand. The promoter is said to be located towards 5' end upstream of the structural gene. It is a DNA sequence that provides binding site for RNA polymerase, and it is the presence of a promoter in a transcription unit that also defines the template and coding strands. By switching its position with Terminator, the definition of coding and template strands could be reversed. The terminator is located towards 3' end downstream of the coding strand, and it usually defines the end of the process of transcription. When RNA polymerase reaches the terminator sequence, transcription stops. The newly synthesized RNA strand is released and the enzyme detaches from the DNA. To summarize, transcription begins at the promoter, continues through the structural gene, and ends at the terminator. The template strand is used by RNA polymerase to synthesize RNA, while the coding strand has a sequence similar to RNA. This process ensures accurate transfer of genetic information from DNA to RNA. Now it's time to test your understanding. Try these important questions. These questions are important for your CBSE and NEET preparation, so practice them carefully. And if you found this video helpful, like the video, share it with your friends, and subscribe to the channel for more biology concepts, important questions, and exam preparation videos. Keep learning, keep practicing, and keep growing.